Morning, everyone. I'm described as a genomic futurist. That's a title that not many people have. And it's actually a little strange because the future, of course, isn't here yet, so no one really understands it. And of course, genomic technologies and cell technologies are at a scale that no one can see. So it's actually really difficult to go and describe this type of work to my friends and colleagues and peers. So what I want to do is actually just take you a little bit into my world. We're all used to technologies and how quickly things change these days. In fact, it's what's really powering a lot of what we're talking about here at BIF. Things like cell phones, the internet, most other technologies that are allowing us to link together. These are exponential technologies, and they change very quickly. But there's an exponential technology we take completely for granted, and it's the living cell. It's the fundamental unit of life. We all start off as a single cell, and we grow to become a very powerful, coordinated network of hundreds of trillions of cells. Layers and layers and layers of different systems architectures that we think of as organ systems, our brain, our lungs, our liver, etc., all working together to create this powerful mechanism. We don't understand this mechanism very well. In fact, take it all down, right down to the molecular level, and the living cell is still almost completely unknown, a mystery. It's a marvel of nanotechnology. I've been studying cells for over 25 years. I didn't start with the human body, far too complicated. I started with bacteria, and they're my first love. The bacterial cell has been around for almost 4 billion years. They kind of look like little hot dogs. All they do is they grow and they eat and they kind of separate like sausage and they go off their separate ways. And they're some of the most powerful organisms on the planet. Every single cell in your body is actually powered by a little bacterium that it made a relationship with billions of years ago. Bacteria are digesting your breakfast now and presenting you with that energy. Bacteria were here long before you, and they'll be here long after you. That being said, bacteria and every single one of our cells has mechanisms that we're starting to understand. I got into genetic sciences in 1990. I was really fascinated by how we could read the genomes of these cells, literally the operating system, and figure out from that code how these cells worked. You might remember in 1990, the Human Genome Project got started. It had taken 5 years to get that project underway. 5 years of putting together the resources and the scientific intellect and will to start reading a human genome. What they didn't tell you at the time was that there had only been one genome sequenced to that date, a little virus. They hadn't even sequenced a bacterial genome at that time. So for the next several years, I started organizing genomes, and they're fascinating. They're maps of literally metabolism and how things work. It's code. Within a decade, we completed not only bacterial genomes, but the human genome. That was published in 2000. And we still don't know that much about life. We really don't. In fact, it's been, in the ensuing 12 years, there's been papers written where people have said, this genomic stuff hasn't really changed our lives, has it? We tend to underestimate, though, the power of these technologies because the genomic wave is starting to come, and that's what I'd like to tell you about. In 2000, I woke up and I realized I had to blow up my entire life. I'd spent the last several years working in the biotech industry, and it was amazing. It was absolutely amazing. I worked with the smartest people. We had virtually unlimited amounts of money. I knew what we were doing was helping people at the end of the day because we really made things that helped them with their diseases. But we couldn't help everybody. And the thing that was really getting to me was that of all the technologies I'd ever worked with, this one couldn't accelerate. Most technologies get faster and better and cheaper over time. And in fact, if you go back and look at 60 years of drug development, there's a consistent decrease in the outputs per $1 billion invested. We actually make fewer drugs for the money over time. And that's not going to change until the systems change. And I knew this. I was working with this incredible company. And I realized, I've got to start fresh. So I did. I sold everything, and I rebooted my life. And I started to connect with people that thought differently. And there was one piece of equipment that I knew that was going to change all this. And it was called a DNA synthesizer. Now, you know that your genomes are made of DNA. It is code. We were actually doing the Human Genome Project reading that code. But there was another little piece of technology that was allowing us to write it. It wasn't very fast, it wasn't very efficient, but it was literally like a printer for DNA. And that, I knew, would allow us to unlock genomic technologies, genetic engineering. Now, you don't make any friends telling people you're a genetic engineer, trust me. You really don't. But when you start saying, hey, here's how you can become a genetic engineer, or here's how your child can become a genetic engineer, then it starts to change the actual conversation. So for the next several years, I went around and I found the people that were developing this DNA synthesis technology, and they wanted to share it. By 2004, it had reached price parity with cloning, the traditional way we worked with DNA, pulling the DNA molecule out and editing it with molecular scissors. And ever since then, it's just becoming more powerful. In 2004, a class at MIT started teaching students how to do this. They started with grad students, and then they said, no, we have to start working with undergrads. Undergrads, the people just coming into university, are like student stem cells. They're undifferentiated. They don't know what they don't know. These guys, there's been 10,000 students through that program since 2004. The technology of DNA printing is getting incredibly good. Right now, you can get 5 bases per dollar of DNA. That's expensive. Imagine writing an essay, paying literally $1 for every 5 letters. You're going to write short essays. But it gets better exponentially. And I've already seen technologies in development that will allow us to write thousands or tens of thousands of bases per dollar, which shouldn't surprise you when you think about it, because every time one of your own cells divide, it has to write a new human genome. Where is this going to take us? I honestly don't know. I know this, though. We have some reference points. It turns out that the living cell isn't that much different than a cell phone. The living cell is actually a type of computer, far more complex than an electronic computer, much more complex. But it fundamentally has an operating system that we can read unambiguously. We're able to reprogram it. We're able to load apps on it. It is aware and sensing its environment. And it self-manufactures. It's green. It's infinitely sustainable. It turns out that all the work that we've done with computers for the last 30 years actually informs us as we become biological engineers. It actually teaches us how we write code, how we share code. We can use all the technologies and understanding from growing the internet and our computing architectures in genomic technologies. And in fact, you can't do genomic technologies without this computer foundation. So you start small. No one sits down and begins programming by writing Windows 8. You start with Hello World. And that's exactly what's going on. I believe this is so fundamental that it's going to radically change everything you ever thought of about life, about the biotechnology industry, and perhaps about health as well. Where I'm starting to see it come into play already is in very simple structures. What we end up doing when we rewrite the cell is we change its metabolism, its program. You've probably heard of biofuels. That's where literally we've added new pathways to a living cell. Usually it's something small, like a yeast or an algae, and we convince it to create some new molecules. In the biotech industry, we tend to make single proteins that can act as drugs. Single proteins are small, they circulate around your body, they have a defined function, they're like toasters. And they either fix that pathway, clean up that infection, etc. So we're already starting to see the biotech industry get a whole new wave of programmers coming in. The bottleneck is regulation. It takes 15 years to get a drug approved. Now you can actually write a new drug or the code for it in literally a few hours. Where does it go from there? It starts to change the way we think about evolution, because we can move far beyond, well, what happens when you start taking control of your own code? I don't know. I know how we've got here. You know, we can go into the bedroom and create new life pretty easily. Today, if you change one base pair of even the simplest organism, it's an engineered product. And I don't know how to get that approved quickly. This stuff is going to spill over the edges of technology and into your daily lives. And you'll start to see it first by the interpretation of genomic code. We hit a $1,000 price point for reading genomes this year. That's more information than any of us know how to deal with in terms of our life and our health and our bodies. The people that'll figure this out are the marketers. They'll start recommending to you different skin creams, different drugs, different dates. After that, we actually have to start thinking about where we want to take this, how it'll shape our world. We've been pushing nature into the peripheries. We've built our cities. Overall, I see this technology being able to integrate life and sustain us as the way forward. We'll start to bring nature back into our cities for building materials, for fuels, for medicines, and ultimately, even to think about how we move forward as a species. And with that, I think that you'll see that connecting, inspiring, we have to do that with our children with these technologies. But ultimately, this is a technology of transformation. We have to think carefully about how we do that. And I know that people like you in this room will help take that forward in a positive way. Thank you.